Today we will be talking about enzymes, how they are controlled, and we will talk about allosteric enzymes. Right here we're going to be looking at ATC right here. So this is actually ATC ACE. So the ACE right here, the ASE, means that it's an enzyme, okay? And what makes this one special is that it is an allosteric enzyme. It is an allosteric enzyme. So we'll just put allosteric enzyme. Okay, so what is an allosteric enzyme? An allosteric enzyme is an enzyme that changes shape whenever a substrate binds to it, okay? So essentially the quaternary structure, this is a quaternary structure, it's just a collection of subunits, okay? This whole structure changes depending on what it's attached to. So for instance, this one right here is going to be a, I believe it is a uncatalytic right here. So this is the uncatalytic form. And what I mean by that is this is ATC ACE whenever it's not binded or bound to anything. Okay, so this is like the neutral ATC ACE. But whenever it binds to something, specifically whenever it binds to ATP or CTP, which is an inhibitor, it will change shape. So the catalytic form actually has, the catalytic form has, well, it has two trimers, okay? So 6 subunits. And of those 6 subunits, it's going to have 2 trimers, okay? Now, if we remember what a trimer is, it's essentially three units joined together, so 3 subunits to make 1 subunit. Right here we have 3 trimers, okay? So this is a catalytic trimer. This is 1, 2, and 3. So that's the uncatalytic form, and we have the dimers. So it's just 2 subunits paired together. That's going to be 1, 2, and 3. So we have 1, 2, 3 dimers and 1, 2, 3 trimers. So 3 plus 3 is 6 subunits altogether. For the catalytic form, which is just ATC ACE when it's bound to something, it's going to have 6 subunits. That never changes. But instead of having 3 trimers, it's going to have 2 trimers. Most likely it will have an extra dimer as well. Okay, so why is that important? Well, it's important because allosteric enzymes, whenever they bind to something, the changed shape affects the reaction. So changed state affects reaction. And we'll just call that RxN for reaction, okay? So for instance, we have these Michaelis-Menten's charts, okay? And we see that normally this is the sigmoidal curve, okay? It's just the data that was given by experiments and without anything attached to it. So in its uncatalytic form, ATC ACE has a pretty normal looking velocity, okay? But whenever you add ATP, which is a substrate, the reaction velocity, as you can see right here, actually increases. So the catalytic form with ATP, so ATP catalytic form, velocity increases, okay? But whenever the ATC ACE is bound to the inhibitor CTP, the catalytic state makes the reaction velocity decrease. So we'll put CTP, which is an inhibitor, the catalytic form, the velocity actually decreases. So no matter what happens, we're always going to go to a catalytic form, but depending on what we touch or what we connect with, our catalytic form is going to change the reaction differently. So if we attach to the ATP, the velocity will increase, which is good. If we attach to inhibitors, the velocity decreases, which is bad. And later on in this video, we're going to go into the mechanics of why inhibitors actually slow down reactions. okay? And so whenever you're working with something that positively affects the velocity, for instance, ATP in this case, we would call that an allosteric activator. So we call these allosteric activators, something that has a good effect on us, okay? So call that allo. And for the rest of this video, allosteric is going to be kind of simplified to allo, okay? So this is allo, this is the allo activator. And something that has a negative effect on the reaction is going to be an allosteric inhibitor. Right? But what is some of the ways that inhibitors can actually stop the reaction from completing or from reoccurring? How can an inhibitor do that? Well, let's ignore this left side. So ignore the little dashed lines for now, okay? Let's imagine that we're doing a reaction in a very normal fashion, okay? So we have enzyme 1, enzyme 2, enzyme 3, etc., okay? So enzyme 1 gets the substrate and makes a product. That product goes to the next enzyme, reacts with the enzyme, makes another product, and it keeps going and going and going until you have the final product, okay? And then that final product just goes on and does whatever it needs to do. But normally, whenever you add an inhibitor, you have a feedback cancellation. So this is called feedback inhibition. That's an H. Inhibition. on. So that's an ION actually. Okay, so what is that? Well, imagine that we continue to do the reaction, okay? But then somehow an inhibitor, it can go on any enzyme, okay? Any enzyme within this chain. So an inhibitor says, you know what? I'm going to go on enzyme 4 and I'm going to hijack this reaction. So now enzyme 4 is inhibited, okay? And it makes the product. But that product goes to enzyme 5 and then it goes to 6, whatever. So it continues the reaction normally. But whenever you get to the final product is tainted. So this is altered, okay? So you can have an inhibitor on step 2, step 3, whatever. If you add an inhibitor, the final product will be altered, okay? And it's altered in the way that the final product will go back to the first enzyme and block the enzyme from attaching to the substrates. So the final product is going to prevent the reaction from reoccurring, okay? So that's how feedback inhibition occurs. You have an inhibitor that attaches itself to an enzyme, and then it makes a final product that is altered, and that final product cannot, it's going to go to the first enzyme and then mess with it, okay? So that's kind of how inhibitors work, but they can also do some other stuff, and we'll cover that later in the video. But right now, this is kind of like the basic idea of what inhibitors do. When we're talking about allosteric enzymes, so allosteric enzymes, we can't really use the same ideas that we used for Michaelis-Menten's enzymes, okay? We can kind of use the same concepts, right? So velocity, substrate concentration, etc. But when we talk about affinity, so the K values, we cannot say, oh, you know, the affinity for this enzyme to this substrate is different. No, that's a Michaelis-Menten's concept, and that does not apply to allosteric enzymes. Instead, we're going to be using K-half, K.5, okay? So this is 0.5 right here. Let me just redo this 5. But why is that important? Well, it's important because we actually have two types of allosteric enzymes, okay? So within the allosteric enzymes, there are two types, and the first type is called a K-type. So we have the K system, and essentially it's when inhibitors they change the K5 value. Okay, so inhibitors change the K05 value. So what is the K.05 value? Well, this one right here is essentially the concentration of the substrate whenever we are at 1/2 the velocity. Okay, so it's essentially, how should I say, at half the velocity, the maximum velocity. So actually it should be this one right here. So half of the velocity, the amount of substrates available changes. So let's put that all together. For the K system allosteric enzymes, inhibitor change the value for K05. And K05 is just the value of the substrate concentration at 1/2 the maximum velocity. Okay? So at 1/2 the, let's make an example. So at half the maximum velocity, let's say that normally we have about 10 substrates. Cool. But in a K system, whenever you introduce the inhibitor, the substrate concentration, instead of being 10 substrates, it'll be like 2 substrates or 3 substrates, etc. Okay? So it changes the amount of substrates available whenever the velocity is cut in half. Okay? So for the K system, we're going to be having the, well, not K system, but we have a V system as well. Okay? So we have the K system and we have the V system. And the thing is, inhibitors actually change, so they change Vmax value, but the K.05 value stays the same. So it's kind of like the inverse of it, right? So K system, they have a change of the substrate concentration whenever you have half the maximum velocity, but for the V system, the K05 value stays the same, so we still have 10 substrates, but the rate of the reaction is altered. Okay, so V system, the velocity changes, right? But the K system, the K value changes. So this one decreases, this one decreases as well. Well, it could decrease or increase, but they are the ones that change. So on the exam, if you have a question that says, in a K system, what value changes? You would say, okay, well, at half the maximum velocity, the K system will have a change within the substrate concentration. If on the exam they ask you, okay, well, what changes in a V system? Okay, well, the velocity stays the same. Okay, actually the velocity changes, but the K05 value stays the same. And you could just tell by the letters right here. So that's pretty simple. We will now be talking about homotropic effects and heterotrophic effects. So homotropic effect is what happens to an allosteric enzyme whenever whenever 2 or more substances bind to the enzyme. Okay, so kind of like interactions between 2 or more, and we're going to say substrates as S, 2 or more S, with the same enzyme. So an example of this, let's say that we have a box, right? So that's an enzyme right here, and we're going to have 2 little triangles attached to it. Okay, so these are the same substrates. That's called a homotropic interaction. So we have 2 of the same things or more. There could be like 4 triangles attached to the enzyme. And whatever effect that is specific to that interaction is called a homotropic effect, right? So let's say that whenever you have 2 triangles, your blood thins a little bit. That's called a homotropic effect because your blood only thins whenever you have two or more triangles attached to this box, okay? If there's only one triangle, well, your blood is not going to become thinner because you don't have a homotropic effect. You just have a normal binding, okay? So sometimes your body reacts differently to enzymes that are having homotropic effects within their substrates, okay? So that is pretty cool. But let's talk about heterotropic effects. And essentially it's the same thing. But instead of having two of the same things or more, you have two different things. Same thing. Let's not use a semicolon, but with different molecules. Okay, so let's use our box again. And right now we're going to have a triangle and maybe a circle. Okay, so now we have two different substrates attached to our enzyme. Now we can have different reactions. Let's say that whenever you have two different things right here on this enzyme, you produce more ATP. Let's just make an example. Whenever you only have a triangle, you make the same amount of ATP, like 5, right? Whenever you have a circle, you make, again, 5 ATP. But when you join them together, let's say you make 10 ATP. The increase of ATP is called a heterotropic effect, right? So because you have two different substrates attached to the same enzyme, you have a different outcome, okay? Whenever you get a different outcome from the same substrates attached to the same enzyme, that is called a homotropic effect, right? So here's a real-world example. Remember when we were talking about the ATCase, right? And we were like, okay, well, you know, whenever ATCase attaches to ATP, you have a increase in velocity. Okay, but what if we attach ourselves to CTP? We're going to have a decrease in velocity. Now, tell me, is ATP different from CTP? I mean, they have different letters, so yeah, they must be different. So the increase or decrease in velocity is actually a heterotropic effect. Depending on what you bind to, you're going to have a different outcome. If you bind to the ATP, you're going to have an increase in velocity. If you bind to the CTP, you have a decrease in velocity. So these are two different substrates with two different outcomes. That is a heterotropic effect. Now we will be discussing concerted models, right? So what is a concerted model? Well, in the allosteric enzyme world, there exists two states an enzyme can exist in, right? So we have the R state and we have the T state. Now the R state is actually relaxed, okay? So this is when substrates are present. So substrates are present, okay? And this one is when the enzyme is tight. And this is when substrates are not present. So let's say that over here we have a square, okay? We have a square. And notice that this square has no openings for the enzyme, okay? So this is going to be the tight state. So the tight state, it's not going to have any active sites for the substrates to bind with, right? And even if, let's say that somehow a substrate sneaks up on an enzyme, it's not possible, but let's pretend that it is. If a substrate tries to connect to a tight enzyme, the binding is going to be very, very weak, okay? So the binding of a tight enzyme is very weak. Cool. But whenever we have a relaxed enzyme, we have something like this. So notice that there is something for the substrate to connect with. And let's say that we have a substrate that connects, and that's good. So, right? So this is going to be the R enzyme. So it's the same enzyme, right? But whenever a substrate comes around, the enzyme relaxes and creates active sites that the substrate can connect to. Now, whenever this happens, the binding of the substrate with the enzyme is very strong. It's very, very good. Okay, so there we go. But now, let's talk about what makes it concerted. So normally in a system, let's say that we start a reaction. Well, we have a system that is new. So there are no substrates within the system yet. So all the enzymes in the system are in the T state. They're tight, they're sleeping, they don't want to do anything. But we have to add substrates into the system for the reaction to get going, right? Because if you remember, enzymes plus substrates give you the ES complex, which gives you the product, right? So right now, we only have enzymes, but we're going to have to add the substrates later. Whenever there are no substrates around, the enzymes are in the T state. Okay, cool. So in order to get this reaction started, we're going to always have to put in some substrates. That's the rule in science. Here's an example, right? So whenever you wake up, you're in the T state. You're tight, you don't want to do any reactions, you don't want to get to work, you don't want to drive, you're tired, you just woke up. So you're going to have to add something to your body, to your system, in order for you to wake up and get into the R state, the relaxed state. In this case, it would be coffee. Let's pretend that the coffee is a substrate, right? So you didn't have any coffee in the beginning when you woke up. But when you get some coffee in your system, all of a sudden, all of your body wakes up and you're ready to go to work, ready to cry, you're ready to eat something. You're going to have a great day. Okay. So what I'm trying to say is, whenever you add a substrate to the system, all the enzymes in that system automatically go from the T state to the R state at once. Okay. So let's do this in purple. So let's say that the T goes to S. So this little line right here is going to be a shift. So this is called a S shift. Actually, not S, but rather the R shift. So that was kind of weird, but let's go to T to R, and this is a R shift. So in the system, all the T enzymes shift into gear, and they become the R enzymes whenever you add substrates, okay? So that's what makes it concerted. Concerted just means that it happens at once at the same time. Now we will talk about affinity. Okay, so we know that affinity is usually defined as K, but remember that affinity is kind of a Michaelis-Menten concept, and remember that we can't apply those concepts whenever we're talking about allosteric enzymes. Instead, we're going to be talking about the affinity for the different states. So this is the affinity of the enzyme to the substrate within the relaxed state. Okay, so that's the Kr value. So enzyme love. So love in this case is affinity. It's just the wanting to attach to something. So enzyme love for substrate when in R state. So relaxed state. Okay, so now we can actually predict the next affinity factor. So right here, this is going to be Kt. So it's the same thing. So the enzyme love for S when in tight state. Okay, cool. These two, in textbooks, you'll often see them as dissociation constants. Okay, so these two terms are called dissociation constants, right? So I think that's pretty cool. We can actually make another equation, and we can say that C is going to be the ratio of the dissociation constants, right? And you'll see why this plays a huge factor. So we have we have Kr divided by Kt is going to give us C. Okay. So what does that tell us? It tells us that, well, if we have more love for the enzyme or for the substrate whenever we're tight, we're going to have a very, very small C value. But if the enzyme wants to attach to the substrate in the relaxed state, which it usually does, the C value is going to increase. Okay, so that's kind of like a mathematical representation of what we just wrote. We will now be talking about the L value. So what is the L value? L value is essentially it's the calculation between how many T enzymes are present in the system divided by how many R enzymes are present in the system. So here's the L value, and if we have more enzymes in the T form than we have enzymes in the R form, the L is going to go really, really up. Okay, so that's not good. Why is it not good? Well, if you have more enzymes in the T state than you have in the R state, the substrates cannot attach themselves to the enzymes and they cannot make products. So do you want the enzyme to be in the T state? Not really, no. Because if you have that, you're not going to be making products. And so a large L is unfavorable. So we could say that large L is bad, okay? You actually want to have more R enzymes, okay? Small L is good. Okay, so there you go. But let's kind of bring out a chart to really solidify the idea. Here is a chart for our values right there. So notice that in the L, so this is going to be the L section and this is going to be the C section, right? So let me just write it here, L, and this is going to be the C section. If we have more T enzymes than we have R enzymes, the L value is going to be increased, right? So notice what happens when I increase the L values. I go from a value of 1 for the first line right here, so this is a value of 1, and this is a value of 10,000. Okay? So notice that whenever the L increases, the overall velocity decreases. So right here we have velocity. We'll just put it going upwards, we have velocity. Whenever I have a very small L, the velocity is going to increase a lot, okay? Because that's favorable. You want to have a small L. What does that mean? Let's use numbers. Let's say that T is equal to 1. And let's say that R is equal to 0.00001. If you put that in a calculator, that's going to be a very large number. Actually, yeah, so you don't want that, right? You don't want to have more T enzymes than you have R enzymes, because then you're going to have a very large number. You're going to have a big L. But let's imagine that T is equal to 0.001, and then R is going to be equal to 1, right? So we have 0.001 over 1, and that gives us an L value that is very, very small. Okay? So that makes the velocity increase, and that's good. But now let's talk about the C value. So the C value is different. The C value, you want it to be, well, you want it to be very large. Why? Well, the C value is equal to the affinity of the enzyme to the substrate in the relaxed state divided by the affinity of the enzyme to the substrate in the tight state. Okay? So what does that mean? Essentially, you want the affinity for the enzyme for the substrate in the relaxed state to be high, right? So let's say that Kr in this case, let's say that Kr is equal to 50. Let's do it better, that's 50. And let's say that the Kt is equal to 1. So 50 is a pretty good number. That means that the enzyme wants to go with the substrate whenever it's relaxed. And then a KT of 1 is pretty good too, because the enzyme doesn't really want to be with the substrate whenever it's tight. So let's put that in here. We have 50 divided by 1 is going to be 50. Okay, so C would equal 50, and you would have something like this. Okay, and the velocity is going to go really, really fast. Cool. But what happens if Kr is equal to 1 and then Kt is equal to 50? You would have 1 divided by 50, which is very small. So you would have something like this. Notice that the velocity would actually decrease. Not as good. Okay? So what's the best case scenario? The best case scenario is that you have a very high Kr value, and then you have more R enzymes. Okay? So what does that mean? It means that in your system, you have a ton of relaxed enzymes that have a lot of love for the substrates. Whenever you have a lot of enzymes that want to react, you're going to make a bunch of products really quickly. And it adds up very nicely, conceptually. Okay? So that's essentially the concerted method with L values and C values, and also the dissociation constants. And before I forget, so the reason why inhibitors limit the amount of product you can make is because inhibitors actually stabilize the T enzymes. Okay, so whenever enzymes are in the T form, inhibitors come in and they say, hey, enzyme, you're going to stay tight, you're not going to accept any substrates, and you're not going to make any products. And the enzyme is like, okay, cool, it's a day off. So whenever a substrate tries to attach itself to the enzyme is not going to react with it, and no product is going to be formed. But activators, allocentric activators, they actually go to the enzyme and say, okay, well, you're going to be relaxed, you're going to stay relaxed, and you're going to bind to the substrates, and you're going to make product for me. Okay? So activators actually keep the relaxed enzymes stable. They keep them open up so that they can do reactions and products. Inhibitors stabilize the tight enzymes, and they keep them tight so that they cannot do reactions with the substrates and they cannot make products. We will now be talking about sequential models. Okay, so let's imagine that we have a T enzyme and it's in a relaxed state. So let's just put sequential first. What's going to happen is that the enzyme is going to bind to, is that the substrate is going to bind to the tight enzyme, and it's going to do a series of reactions that converts half of the enzyme into a relaxed state. Okay, so here's an easier way to imagine it. Let's imagine that an enzyme is a huge unit. Okay, and this huge unit in this case has two subunits, right? So we have one half of the subunit is a T, and the other half of the subunit is a T. So whenever these subunits merge together, they make the enzyme. Now when the substrate connects to half of the subunit, it becomes an R state. And then the R state kind of convinces the T portion of the enzyme, okay, to become R as well. Right now, this is called a cooperative binding, okay? So let's go over it again. The substrate goes to one of these subunits, and it says, okay, well, you're not going to be tight anymore, you're going to be relaxed. So now the subunit becomes relaxed, and then the subunit is going to look to its friend, which is another subunit, and it says, hey, since I'm relaxed, you should be relaxed as well. And then the tight subunit says, okay, I'm going to be relaxed. And overall, the enzyme becomes completely relaxed. Okay, so this is what it means to be sequential. It doesn't happen at once, right? In the concerted model, whenever a substrate comes into contact with the enzyme, this becomes relaxed and this becomes relaxed automatically. But in the sequential method or model, the substrate goes near to the enzyme, and it takes a little bit for the enzyme to fully convert into an R, okay? But now let's talk about the other binding method. Okay, so this is going to be a negative binding. So this is negative binding. And essentially, it's when an inhibitor goes into the enzyme, okay? And it says, well, I know you're tight right now, but I want you to remain tight. I'm going to stabilize you so that you remain in this form and you cannot make any products. And the enzyme says, okay. And now that they're connected, this subunit right here is going to make it a lot harder for this subunit right here to accept the substrate. It's going to change the activation sites. So notice that we have kind of like a trapezoid shape right here. Notice that it looks like a Batman symbol, okay? Looks like a little bat right now. So it was saying, okay, well, since I'm tight, I'm gonna make the whole enzyme tight. I'm gonna mess with the active sites and change the shapes. So now the substrate can't even bind to the other side. It's gonna take a little bit of time for me to do this, but overall, the enzyme is going to be inhibited, okay? So as a recap, for negative binding, the inhibitor goes to one of the subunits, messes with it, and then that tight subunit is going to influence the other subunit to be tight as well. And overall, it's not going to make any products because now the enzyme is fully inhibited. It just took a little bit longer because it happened in a sequence, okay? It did not happen at once. Now, notice that for the sequential model, we actually used an induced fit method, okay? This can also be called the glove model, okay, from the last enzyme video. So as long as the inhibitor or the substrate has kind of roughly the same shape as the active site, it's going to go in there and react. What happens when you want to convert an enzyme from a T state into an R state? Well, let's say that you want to do it differently. You don't want to put any substrates in it. Okay, fine. But here's a key. Like, here's a trick that you can do. You can actually treat enzymes with hydroxyl groups, okay? So adding OH to enzymes shifts shifts T to R, okay? So it makes it active. It's always gonna be T to R, okay? Only with ATP, okay? Because essentially what we're trying to do is we're trying to phosphorylate the enzyme, okay? So we're doing a phosphorylation, okay? So we're going to be using the ATP phosphate, and we're going to be using the hydroxyl group. Where do we get the hydroxyl group? You actually get the OH from serine, threonine, and tyrosine, right? So let's make an example. Let's say that in the lab you have a sample of T enzymes, and then you're giving some ATP. You can decide to put in some serine, some threonine, or some tyrosine into the test tube with ATP. What's going to happen is that the protein is going to connect with the serine molecule, okay? So you remember how in serine we just have R and then we have the OH, right? The protein is going to connect to the R group and the OH, and then the ATP is going to connect to the hydroxyl group, okay? So pretty cool, right? So whenever that happens, the bond, ATP is adenosine triphosphate, okay? So that means that you have 3 phosphate bonds. Whenever one phosphate bond breaks, it releases a bunch of energy, okay? And that energy is going to be used to convert the enzyme from a T state to an R state, okay? That's how ATP is used in the body. So you can do that with threonine, tyrosine, or serine, and that's all that happens. I think it's pretty cool. We will now be talking about Zymogens. So what is a Zymogen? A Zymogen is essentially an inactive enzyme that can have some of the bonds cut. And whenever you cut some of the bonds, depending on where you cut. You can make the inactive enzyme, which is in the T state, into an active enzyme, which is in the R state. Now, this process is irreversible. That means that you cannot undo this. Okay, so irreversible process. Okay, and in this example, well, if you're working in the lab, you're most likely going to be using chymotrypsinogen. Okay, so right here, we're going to have chymotrypsinogen, right? And it's going to be cleaving the arginine. Well, let's hold on. So in this example, we're working with chymotrypsinogen, and we're going to be using trypsin to cut the arginine group in the chymotrypsinogen. Sorry. Okay, so now whenever you cut that, you're going to have some reactions going on, and it's going to keep getting cut and cut and cut again and again. Essentially, you're going to have the alpha chymotrypsin as a product, and this is going to be the active enzyme. So what am I trying to say? I'm trying to say that first we have an inactive zymogen. A zymogen is an enzyme. In this case, chymotrypsinogen is going to be the enzyme. So it's an enzyme that has the possibility of getting cut into an active species. Okay, so we can treat chymotrypsinogen, which is trypsin, and it's going to do the cutting. Okay, and it's going to keep cutting until we have the active species. So again, this is an irreversible process. And so now you have kind of like different methods to make inactive enzymes into active enzymes. So one way that you can do it is that you could treat it with trypsin, right? Or you can just convert it using ATP and hydroxyl groups. So that's how you convert inactive species to active species. So hopefully now you understand how to calculate the L values, the C values. You know what that means. You now know what a zymogen is and how enzymes can exist in the T state and in the R state, right? Hopefully this video helped out. I know that you'll do well on your exam, and thank you so much for watching my videos and for spending your day with me. I hope you have a great day and that you do well on your exam. So thank you, and take care.